All right, so hope you had a good weekend, and today we're going to start this process of looking at how plants go from basically being a germinating seedling to an adult plant, and how they can acquire, distribute, and store resources, and do all of this while they're basically anchored in one place. So there's a lot of differences between plant and animal development that you've been introduced to in lab to some extent, I realize, and we're going to look at that in a lot more detail here in lecture. So start off looking at seed germination, and then we need to review some cell and tissue types to give you some orientation there before we get into looking at how the plant body grows and develops. We'll start out today looking at the shoot system and get to roots next time. So I did post some questions online yesterday. You should have got an email about that. Some sample questions, give you just a bit of an idea of the kinds of questions to be asked during the midterm. And the midterm is going to cover not all the material in this part of the course, but up to the end of this week, there might be a little bit of material from the beginning of Monday's lecture, depending on how far we get, but I'll make it clear where that breakpoint is. And then the last 2 lectures or so will be on the final, but not on the midterm. Final will potentially cover all the material in this part of the course. All right, so last time left off by showing you an example of a eudicot and a monocot seed and how different these can be morphologically, anatomically. And I pointed out how the endosperm, which is the nutrient tissue for development of the embryo in angiosperms, is absorbed by the cotyledons, those embryonic leaves, in many eudicots. And so at the time that the seed is actually mature and goes dormant, there isn't any remaining endosperm that can be found inside the seed. It's all been absorbed by the cotyledons, and the cotyledons then are the source of nutrition for the germinating seedling. Whereas in the monocots, there is endosperm left, and it actually constitutes a large amount of the contents of the seed. And the cotyledon, of which there's only one, as the name indicates, serves as sort of conduit for absorption of those nutrients into the embryo during germination. And I just want to make a point about the morphology of grains, which are basically the fruits of grasses, as relates to our own diets, because of course a big part of our diet is our grains. A huge proportion of our carbohydrates come from grains, and the milling or refining of grains results in the removal of some of these contents of the seed and fruit here that can render them much less nutritious. You've heard of whole grains. Whole grain is basically the entire fruit, all of its contents in a commercial sense. Any kind of whole grain cereal or whole grain that you buy is the entire fruit. Remember, the fruit wall is fused tightly to the seed here. They're not really extricable from one another. But when you mill or refine grain, all you're left with in the end is just the endosperm. So white rice, various milled grains are just the endosperm. Also, when you pop corn, that white fluffy substance is the endosperm, for example. And it contains a lot of carbohydrates, but the bulk of the nutrients are actually in the embryo, proteins, vitamins, etc. The embryo is commercially known as the germ, and you've no doubt heard of wheat germ, that sort of thing. That's removed during milling or refining, as is the bran, which is the fruit and seed coat, which is the main source of roughage, important in digestion. So those are some of the features of the grain fruit that are important commercially. All right, so germination then. I mentioned last time there are a lot of different cues that can be involved in initiating germination depending on the plant species. But in all cases, it involves absorbing water, which will cause the contents of the seed to swell, bursting the seed coat, and germination is initiated. But how exactly the seedling forms from that germinating seed can vary a lot between eudicots and monocots, for example, and even within each of those groups. And we're just showing a couple of examples here that are in your textbook to show you how different this can be. So in the case of the eudicot bean example here, so in both cases here we have the seeds, or here the fruits underground. And in both cases, the embryonic root emerges first during germination. So getting a root system established is job one for a seedling. It has to be able to acquire water before it can start producing aerial parts that are exposed to the atmosphere and desiccating air. And so we have this embryonic root, the radicle, that basically has positive gravitropism. Tropism means a growth response, and positive gravitropism is basically growing in the direction of gravity. And so the taproot, which is the descendant of the embryonic root, the radicle, grows downward, starts to branch. And the same thing happens in the monocot grain here. We have the radicle forming a new root, although the taproot from that radicle doesn't persist very long in monocots, as I mentioned earlier. These have a fibrous root system. The taproots are pretty quickly replaced by adventitious roots that are formed from the base of the stem. But in any case, positive gravitropism here again with regard to the growth of the roots. Now, in the eudicot, that stem region below the point of attachment of the cotyledons is called the hypocotyl, which means below the cotyledons. It has a negative gravitropism, and it actually starts elongating, forming what's called a germination hook here. And as it elongates, it pulls the cotyledons and the embryonic axis that they're attached to, which is protected between those cotyledons which are folded over it. It pulls them above ground. And by doing that, that embryonic axis, which is pretty fragile, is protected. If it was pushing those cotyledons up through the soil, they might spread apart, tear apart the embryonic axis. But by pulling it up like this, the seed coat takes all the abrasion. The outside of the cotyledons can get abraded, but that doesn't hurt them. They have a really good response to wounding. And eventually, they're pulled free of the soil. The whole thing rights itself. Cotyledons, you can see, start greening up immediately. So they're not only providing nutrition that was stored there by the absorbed endosperm to the developing seedling, but they're also photosynthesizing and producing sugars that are important for the development of the new seedling. And then we have the enlargement, the growth of these first foliage leaves, which were tiny inside that seed to start with. And we have the lengthening of the epicotyl here, and the whole thing is on its way. In the case of the monocot, it's different with regard to the emergence of the shoot out of the soil, the stems and leaves, the stem and attached leaves. The coleoptile, which is present in the embryo, is a sheath that surrounds the shoot apex, the embryonic shoot that's going to form the stem and leaves. And that grows straight up through the soil, taking all the abrasion, but it's not a persistent structure. Its only function is to emerge out of the soil, and then the shoot comes up through that sheath, the emerging young stem and leaves, and begin to expand and grow. So you can see in this case, the only cotyledon remains underground inside the seed. It never emerges from the seed. And the bulk of the seed remains underground and eventually disintegrates underground, decomposes, whereas the bulk of the seed here is actually projected above the surface of the soil during germination. So really different ways of germinating in the case of eudicots and monocots. All right, so I'm going to show you now a time lapse of eudicot germination so you can see this process in fast motion. And these are beans just planted under the surface of the soil. And here you can see the radicals growing down. These are root hairs here that help absorb water. We'll talk about them later. Here you can see that germination hook, the epicotyl. And now, the hypocotyl was growing forward, upward. The cotyledons have expanded and greened up here. These are still cotyledons. And we have the first foliage leaves starting to expand down here in between the cotyledons. You can see them here as well. And here they are. They're cotyledons, you can see, are important here for early photosynthesis. And here you can see the foliage leaves now, which have more conspicuous venation. And the whole thing's on its way. You can see the root system is already extensive by the time the shoot has fully expanded there. So now look at what goes on with the monocot again. This is some corn kernels. And here you can see, again, the radical forming, growing down. The young root. Here comes that coleoptile, that sheath, growing straight up. You notice the seed's still underground. And here now we have the emergence of the shoot out of that sheath. And you can see that we have adventitious roots starting to form here. And now you can see the leaves expanding and additional leaves being formed here as it grows. So pretty different growth between just these two examples. And there are a lot of variants and variations on germination across different flowering plants. All right, so now so any questions about germination before we go on here? Okay, so to really understand the development of the plant beyond this point, we're going to have to talk about plant anatomy, which is really fascinating and very different from that of animal anatomy. But like animal anatomy, it involves tissue systems and organs that we're going to get into, and you've been introduced to already in lab to some extent. And all these anatomical innovations are really important to how the plants acquire, distribute, and store their resources and get to be large plants. Here are the tissue systems in different colors over here, from cross-sections of a leaf, a stem, and a root. So these three different cross-sections show the three different tissue systems. And a tissue system is just a functional unit of cells that connect all the organs, of which there are three types in plants. So there are three types of organs in general in the broadest sense. There are leaves, stems, and roots. And remember, the parts of the flower are leaf homologs, so those qualify as leaves as well. So when you boil it all down, there are really three types of plant organs. And those three different types of organs have or to some extent at least, have quite different arrangements of their tissues within them, as you can see here. So there are three major types of tissue systems in vascular plants. There's the dermal tissue found around the periphery, the outside of the plant in blue. There's the ground tissue shown here in yellow, and then the vascular or conducting tissue shown here in purple. And even though the arrangement of these tissues within each of the three types of organs can be pretty different, especially with the vascular tissue, there's seamless interconnection of these tissue types between the organs. There's a really short zone where these organs come together, where their anatomy transitions. So these are continuous tissue systems throughout the plant. So we're going to look at each three of these systems to start with, and we're just going to look at them in the primary plant body. So this is prior to any secondary growth or thickening growth that we get in woody plants. So this is just what we see in herbs and in young growth of woody plants. So the dermal tissue is the outer covering of the plant body. In the primary plant body, it's only one cell layer in thickness throughout the plant. And on the aerial parts of the plant, on the leaves and the stems, these cells, actually the dermal tissue on the primary plant body is called epidermis, which refers, the epidermis is just this outer covering of single layer of cells, tightly compacted. As you can see here, they fit together very tightly and are covered by a waxy surface of that's called the cuticles, we've already mentioned, preventing water loss, as well as, to some extent, for protecting the plant from attack by living agents. And there also then are pores that allow for gas exchange. And these pores shown here are regulated by cells that can open and close them. We'll talk about that later. The pores plus the cells that open and close them are known as the stomata. Stoma is singular here. And these pores are crucial, of course, because the plant needs carbon dioxide from the atmosphere in order for photosynthesis to occur. And it has to release the oxygen that it produces during photosynthesis that's not needed. That is, release what's not needed for respiration. But it also potentially suffers water loss. It's an inevitable consequence. When you have saturated conditions inside the leaf and a much drier atmosphere, you're going to lose water through evaporation to the atmosphere. That's a process called transpiration. And we'll talk about it more later. So this regulation is really important, to let gases in when they're needed, but not let too much water vapor out. And that's, of course, an important adaptation of land we talked about earlier. Also, the epidermis on the shoot, that is on the leaves and the stems, can have epidermal hairs, as they're called, or trichomes, which can serve a wide variety of functions. So their primary function is protective, and they can protect the plant in various ways. They can protect it from herbivory, so they can actually produce noxious compounds. There's a lot of glandular exudates that come out of these hairs, which can be unicellular or multicellular. This isn't a glandular one shown here, but they can actually have a chemical defense. They can have physical defense. They can actually just form a barrier to herbivores. They can also increase the boundary layer, the dead airspace between the surface of the leaf and the atmosphere. They can reduce the rate of diffusion, which can be important in limiting water loss in dry environments. And they can also form a really dense covering in some cases and be highly reflective and prevent the leaf from overheating, which can be a really important thing in hotter, drier environments. So there are a lot of different functions that those epidermal hairs can provide, and in roots they have different function we'll talk about later. All right, so the next major type of tissue is called ground tissue, and this is basically just inside the epidermis in the primary plant body, and it's basically everything inside the epidermis except for the vascular tissue or the conducting tissue, which is shown, the conducting tissue or vascular tissue shown in purple here, the ground tissue shown in yellow. And the ground tissue, as you can imagine, being such a large part of the plant body, has a lot of different functions. And these include the most metabolically active cells in the plant, the ones that are most actively involved in photosynthesis, and also they're involved in storage of starch, for example, and also to some extent cells that are involved in support of the plant body against gravity. There's also function in that support function in the vascular tissue we'll talk about in a moment. And this ground tissue, in eudicots at least, where we have this distinction between where we have the vascular bundles arrayed in a ring like this, there are two zones of ground tissue that are referred to: the cortex that's to the outside of the vascular tissue, between the vascular tissue and the epidermis. That's the cortex. And to the interior of the vascular bundles, that ground tissue is often called the pith. And as you can see, there is no pith in a root of a eudicot. All right, so when we go to the different cell types that make up ground tissue, there are three major cell types. By far the most significant one in terms of overall composition of the ground tissue is the parenchyma. And this type of cell is living at maturity, which might sound strange, but you'll see what I'm talking about in a moment. And they're the least differentiated types of cells in the ground tissue. They're most similar to embryonic cells. And like embryonic cells, they have very thin primary cell walls. And they're quite flexible at maturity. They also are the most metabolically active cells within the ground tissue. Photosynthesis cells that undergo photosynthesis are types of parenchyma, for example. And you can see these green chloroplasts inside these parenchyma cells here undergoing photosynthesis. Being the least differentiated type of cell in the ground tissue, they also have the capacity to de-differentiate. That is to go back to an embryonic state if there's wounding of tissue. So if the tissue is damaged, disrupted, or wounded, parenchyma is the type of cell that can de-differentiate and undergo cell division as a part of a wound response. And so we can have repair involving the parenchyma. And those cells are also important in storage of starches and stems and roots, in addition to photosynthesis in leaves and to some extent stems. Okay, the second major type of ground tissue is called parenchyma. And this is a type of cell that's important for support, physical support against gravity of the young plant body or the primary plant body in regions that are still undergoing elongating growth. So in the parts of the plant body that are still growing, parenchyma can have an important support function. And again, like parenchyma, these cells are living at functional maturity. And they have only a primary cell wall, but it's thicker than the cell walls of parenchyma cells. And you can see this is all primary cell wall between in and around these protoplasts here. And it's irregularly thickened. These cells are generally found just inside the epidermis. So this ground tissue is found just inside the epidermis. And it's important in both stems and in leaves. For example, the petioles or stalks of leaves for supporting those structures. And if you've ever bitten into a celery stalk, which I think almost everyone has, the celery stalk is the petiole or the stalk of a leaf of a celery plant. And those strands that get caught in between your teeth, those green strands are strands of parenchyma. So those are something you can relate to probably, and they're important during those, in those parts of the plant that are still growing. All right, the final type of ground tissue is a type of cell that we find in ground tissue is sclerenchyma. And sclerenchyma is shown here and here in this section. These types of cells are actually dead at functional maturity, which might sound bizarre, but before they die, they basically produce cell walls that have not just primary, but they have not just primary, but secondary cell walls. The secondary cell wall laid down on top of the primary cell wall, and then lignified. So these cell walls, these secondary walls, are impregnated with lignin, that strengthening compound that's highly resistant to decay as well. We talked about earlier. It's a major composition of wood, but wood is from vascular tissue. This is in ground tissue. But like wood, these cells, after they lay down those cell walls, they die. Protoplast disintegrates, and these function in structural support of parts of the plant that have already finished growing. So these cells would be incapable of expanding, or I should say, they're not flexible at maturity, and they only become fully differentiated in parts of the plant body that are already they've grown to their full size. All right, so there are two types of sclerenchyma that's worth mentioning. I'll just mention them because they're important. Sclerids are a short, squat, thick-walled type of sclerenchyma that you're probably familiar with if you've bitten into a pear. That gritty texture of a pear comes from little masses of these sclerids that are inside the fruit of a pear, and they serve a structural function there. They're also really important in the hardness of seed coats and of fruit walls in the case of nuts and other hard fruit walls. So sclerids have an important function in many respects. Another important type of sclerenchyma are fibers. And true sclerenchyma fibers then are elongated cells like this in the ground tissue that have secondary walls. There are also fibers in the vascular tissue that can reinforce vascular tissue, but they're present in sclerenchyma as well. And if you're familiar with hemp fiber or flax fiber, these commercial fibers used in textiles, rope, for example, thread, these are types of sclerenchyma. There are also other kinds of fibers loosely used in many respects, but at least some of the fibers we're familiar with represent types of sclerenchyma cells. All right, so now we're going to move on to the vascular tissue, the conducting tissue. And the sclerenchyma is kind of a nice segue into vascular tissue because not only are fibers, that I just mentioned, also cell types within some of the vascular tissue, particularly the xylem, which is the water-conducting tissue of vascular tissue. But the main functional cells of the xylem are dead at maturity as well and have strong secondary walls impregnated with lignin. So there are two types of vascular tissue. There's the xylem and the phloem, which we'll get to in a minute. The xylem is involved in water conduction as well as conduction of inorganic nutrients from the soil through the roots up into other parts of the plant. And this conduction of water doesn't require energy on the part of the plant. And the cells that are involved are dead at functional maturity. So we'll get into how that works a little bit later, but the two major types of cells in the xylem that are involved in, the main cells that are involved in the actual moving of the water, that actually serve as the conduits for moving the water through the plant,